Sure. We all know during normal PCR method, we use the double stranded DNA and then we melt it and we use the primer and amplify the DNA strand and make the copy millions of copy by the PCR method or polymerase chain reaction method. Now, in real-time PCR, also known as a quantitative PCR, we can analyze in case of a real-time PCR we can monitor the amplification in a real time. That means in case of normal PCR, we analyze the data after we get the data after the end of the PCR reaction. But in case of real time PCR, we get the data simultaneously with amplification. And we can monitor the data with the simultaneous amplification. Now, here we learn about the calculation of fold change by delta city method by using in real-time PCR data analysis. Now let's see in real application of real-time to study the difference in gene expression of a gene in disease state compared to normal state. During we use in real-time PCR during the analysis of gene expression, like in case of also we can use the microarray technique. Microarray also used for gene expression analysis. Here, real-time PCR is much more sensitive than microarray and it give more accurate data than microarray. And in here, in real-time PCR, we use some fluorophore. That's fluorophore emits the amplification fluorophore measures the amplification rates. So higher amount of fluorescence means higher rate of amplification. Two types of basically fluorophores are used. Basically most of the importance are cyber green and the Tachman probe. We will come to later. Now we can see that it's also used for gene expression during cell differentiation or development. Change in expression for cells that are exposed to a chemical substance is a drug, toxin, hormones, or cytokine. Suppose you have given cancer cells with a and you want to know cancer cells with use some anti-cancer drug. Now you want to know that the how much drug efficacy or drugs is working in a particular genes like BCL2 expression level. Now you can compare between the data from that real-time PCR. We'll see in later. Now here in case of cyber green basically bind into the double-stranded DNA and much amount of double-stranded DNA, so much amount of fluorescence. So more amplification, more level of fluorescence. So this way the Tachman cyber green works, right? These two probes fluorophore are actually used. Now here you must have to know some basic information about the real-time PCR. For real-time PCR data analysis, one is the threshold value. One is threshold threshold value or threshold cycle. Threshold cycle also denoted as CT. Another is the baseline. Baseline. Now, during the in case of data analysis, we'll get a graph, and this graph x-axis contain the number of cycle, and y-axis contain the fluorescence. Now, from here, these are baseline. Now, from baseline, so these are the threshold cycles. Suppose it's a 5, 10, 15. So these are 15 is the threshold cycle. So what do you mean by baseline? So is baseline is a so you do mean by baseline? Baseline, the baseline of the real-time PCR reaction. PCR reaction refers to the signal signal level during the initial cycle during the initial cycle of PCR in which there is little change. There is little change. Now above the baseline, these are actual baseline. Now above the baseline, these are the threshold value. So threshold value of the real-time PCR is the level of signal that reflects a statistically significant increase over the calculated baseline. So above the baseline value is the threshold value. So from here is the threshold, and CT, the threshold cycle, is the 15 here. So in that way we can, now we will see how to calculate the data from real-time PCR. Another is the steps of the is here. In case of here, if we use the RNA sample, like isolation of RNA from the culture cells or blood, and then DNA's treatment, because the DNA's treatment is given because it should be DNA free, then we convert it into cDNA by use of reverse transcription method. And we use the cDNA, and cDNA actually lack intron, that is and then we analyze in real-time PCR, determine the PCR efficiency of gene of interest and internal control gene, and presentation of individual data as individual point. Now we'll see the calculation, how we'll calculate the real-time PCR data. Now, real-time PCR data for calculation, we will have to calculate the fold change. So, fold change, we know that fold change is calculated in terms of, so formula is that for fold change, formula is 2 to the power minus delta CT. So, delta CT equal to treated sample minus internal control. And another formula is for the same is that 2 to the power minus delta CT, 2 to the power minus delta CT method. Delta CT method here, CT equal CT delta CT means delta CT equal to CT gene of interest, CT gene of interest minus CT of internal control minus CT of gene of interest minus city of internal control. When when we take two genes and two internal controls is there, and then we can calculate in that way del 2 to the power minus delta delta ct, and in 2 to the power minus delta ct for single gene. Like in case of if we use the suppose in beta cat expression of after treatment, and treated sample is ct value is 30, treated sample treated sample CT for a particular gene or its mRNA CT value, the particular gene CT value is 30, and internal control is 25. So fold change will be for that gene, fold change will be 2 to the power minus delta ct. That means 2 to the power minus 30, that is treated, minus internal control, that is 25. So 2 to the power, or we can say as internal control as 35, we can take it as a 35, so that negative sign will be 1, so 35, so minus, so 2 to the power say 5, so here will be 32 time fold change. Now we'll see another examples how to calculate the fold change in via the delta ct method. Now see that now the these questions actually given the mean CT of the Hoxd gene in treated and untreated sample was for 24.6 and 27.5 respectively. The mean CT of the 18S rRNA, these are internal control, in the treated and untreated sample is 9.9 and 9.8 respectively. What is the fold change in the expression of Hoxd gene due to treatment? Now for that, fold change due to treatment fold change due to treatment equal to 2 to the power minus delta delta ct, right? Now here the now first see that my treated sample, so 2 to the power minus sample ct value of the Hoxd gene treated and untreated sample, so here 24.6 minus, now say you have to consider with internal control. Internal control is generally we use the control like beta actin, but those are housekeeping gene actually used as a reference for that. And every experiment we use as a standard here is that the 18S rRNA is an internal control. Beta catenin like beta catenin also acts as a internal control. GAPDH, beta catenin, so beta catenin, these are basically used beta catenin, GAPDH, GAPDH, so FDH, beta catenin, beta actin, beta actin, and also 18S rRNA. These are different are there. So internal control. So here 24.6. Now these are 24.6 minus 9.9 minus, now here is 27.5 minus 9.8. So here we'll get 2 to the power minus, it will come 14.7. 14.7 minus 17.7. So here we'll get 2 to the power minus, it will come minus 3. So 2 to the power 3 equal to 8. So here, so due to treatment, the fold change is 8 times. Fold change due to treatment is 8 times. So see that the question, read once more. The mean CT of the HOXD gene is given. Is treated and untreated sample was 24.6 and 27.5 respectively. Mean CT, that internal control, that 18S rRNA in the treated and untreated sample is 9.9 and 9.8 respectively, which is the fold change in expression. What is the fold change in the expression of the HOXD10 gene due to treatment? So it will be 8 times. So we have to calculate the 2 to the power minus delta CT method. Here the internal control is 18S rRNA gene as an internal control. So 8 times fold change. So basically the summary, you can say that formula, you have to remember the 2 to the power minus delta CT or 2 to the power minus delta CT. CT is the treated minus internal control, right? Treated minus. So these are the formula for fold change in real-time PCR data. And in real-time PCR data, we can analyze the RNA. Even in this, now the coronavirus, as a coronavirus genome is a RNA, single-stranded RNA. By the most of the detection techniques are based on this real-time PCR. And like in case of here RNA, single stranded RNA is there. Now single stranded RNA, by using the reverse transcription method, enzyme converted into cDNA, and this cDNA actually used for amplification, and then this also, it's by using different Tachman probe or cybergreen probe, we can analyze the expression of a specific gene and into the samples. Like in case of coronavirus, gene, sometimes those are specific primers are based on that. The amplification will be that, like in gene, etc. So these are important techniques for analysis of gene expression. Real-time PCR is important, and real-time PCR also is the very good for actual visualization of the data simultaneously.